Skeletal system is made up of three components, that is cartilage, ligaments and bones. We know there are in a human being 206 major bones in adult human, but tendons are not part of the skeletal system, rather they are part of muscular system or muscle system. So the cartilage we have seen earlier, ligaments we have also seen earlier, and the bones we are going to see. Bones are actually structures that are made up of the connective tissue, collagenous fibers that are spaces are filled with calcium and phosphate. So they are cemented with calcium. These are fibers of protein that are cemented with calcium in order to provide strength and flexibility. Bone are the osseous tissues or supporting connective tissue for the body is made up of three main components, that is periosteum, bone matrix that make about 98% of bone weight, bone cells that make about 2% of the bone weight. We will go through them one by one. Periosteum is the connective tissue membrane, like the perichondrium we saw in connective tissue. The periosteum covers the outer surface of a bone. It has two main layers. The outer fibrous layer, here you can see composed with fibrous layer, and internal one is cellular layer. The fibrous layer is made up of irregular collagenous connective tissue that contain blood vessels and nerves. And the inner layer, it is the layer that is attached to bone, and it is made up of single layer of bone cells, which include osteoblast, and osteochondral progenitor cells, OCD cells. Here, tendon and ligaments, they get attached to the bone. This periosteum, it is replaced by hyaline cartilage, so it is not present at joints. So there, the collagen fibers of the tendon or ligament, they become continuous with those of the periosteum. These fibers, they may penetrate the periosteum into the outer part of the bone, and they become part of the bone. So they strengthen the attachment of the bones with the ligament or tendon. So these collagen fibers that are from tendon and ligament, they are now penetrated into bone. So the periosteum, it is like endosteum. Endosteum is the membrane that is covering bone from inside. Periosteum from outside and endosteum is from inner side. It is same composition to that of the periosteum, but it is made up of only single layer. So endosteum includes the single layer of cells that are osteoblast, and osteochondral progenitor cells. So this membrane is lining bone from inside. It is lining, the endosteum is lining all the cavities. The medullary cavity, that is the main cavity of the bone, and the smaller cavities, the cancellous bone, or the that is called as the fused bone. So these cavities, they are also lined by endosteum. Bone matrix is made up of two main substances. Inorganic substances, they make up to 67% of the bone matrix, and organic substances make 33% of the bone matrix. So inorganic substances, they are calcium phosphate, calcium carbonate, ions of sodium, potassium, magnesium, and the most of these are calcium phosphate. They are about 85%, and calcium carbonate 10%, and then the ions they make about 4%. So these uh, inorganic substances, they are under continuous remodeling, mean they are deposited in the bones, and when they are less in blood, they are depleted or they are uptaken by blood to make their concentration constant in blood. The organic substances, they are strong, they are flexible, they are mostly protein that is in the form of collagen, and the cells. So collagen protein fibers, they make about 30% of the uh, bone organic substances. Then there are a little amount of ground substances that are hyaluronic acid and proteoglycan aggregate. And then cells, bone cells, either they are the osteocytes, osteoclast or osteoblast. They make only 2% of the bone matrix organic substances.
Bone have these four types of cells. The osteogenic cells, they are also called as stem cells or osteoprogenitor cells. So these cells, as the name shows, they are the cells that give rise to osteoblast or other types of cells. So these osteoprogenitor cells, they are located near periosteum, that is the external membrane of the bone, and endosteum, the internal membrane that is lining the medullary cavity. These are the active areas for the production of new cells. So these cells, progenitor cells, they give rise to osteoblast, and they these progenitor cells, they maintain the population of osteoblasts, because when osteoblasts get trapped into extracellular substances and get mature, they become osteocytes. So the osteoprogenitor cells or osteogenic cells, they give rise to new osteoblasts, and the these populations of osteoblasts are necessary for the repairing the fracture in bone and for the bone growth. And the osteoblasts, they are the cells that are that make the bone anew. They are immature bone cells. They are located near periosteum and endosteum. Again, these are the active areas for the formation of new bone. So they are developed from osteogenic stem cell, and they produce new fluid bony matrix, and that is the uh, soft matrix of the bone, that is before the deposition of calcium. So these osteoblasts, they produce the fibrous connective tissue and the hyaline connective tissue, and then later on calcium is deposited. So the this fluid matrix is called as osteoid, that is the matrix of bone before deposition of calcium. Then the later on osteoid are converted into bone when calcium get deposited into these osteoids. So when these cells get trapped into the calcium, they are now get getting mature. They are called as osteocytes. So the osteocytes are most prevalent cells that are surrounded by bone matrix. They maintain contents of bone matrix, that is for the repair when there is damage to the bone matrix. What is bone matrix? It is the calcium phosphate and the protein fibers. Osteocytes are present in bone like the chondrocytes. The osteocytes are also present in depressions in extracellular substance or the uh, spaces. So these spaces, they are called as lacuna. So each lacuna or depression is having one osteocyte. So they are dispersed through matrix in layers. These layers, they are called as lamella, and lamella of the matrix are actually concentric layers of bone matrix, circular bone matrix layers, and they contain osteocytes. So series of these lamella or cylinders around a central canal vessel, they make the structure of the bone. So these cells, the osteocytes, because they are now entrapped in extracellular matrix, that is calcium and phosphate also. So these cells, in order to survive, they have developed special type of uh, their cell growths, or we can say membrane structure. These are called as canaliculi, so that they are attached to other cells through these canaliculi in order to get nourishment. So the cells that are near to the blood vessel, they get food and the gases, oxygen, and then they provide through their canaliculi to the cells that are deep in bone, that are not in direct connection with the blood vessels. So osteocytes, they cannot divide to make new osteocyte. They The osteoblasts are always converted into osteocyte when they get mature and entrap in extracellular matrix. Then we have the osteoclast. They are different because they develop from not from these osteogenic cell, but from the WBCs, monocytes. The precursor of monocytes and these osteoclast cell is same. So these monocyte like cells, they chew like monocyte. They are phagocytic, so they remodel the bones. They help to chew the previous bone so that the osteoblast can lay down new bone. So they secrete proteolytic enzymes, proteases, in order to damage the protein matrix of the bone, and then they dissolve the calcium and phosphate matrix, that is called as osteolysis, and then they release these minerals into blood, calcium and phosphate, and they also help in the regulation of calcium and phosphate in blood. When calcium and phosphate ions, their levels get decrease in blood, these osteoclasts, they become active in bones and they chew bones, and they remove calcium and phosphate into blood in order to keep their level maintained. So the bones are classified on the basis of its shape into long, short, flat, or irregular bone. Long bones are longer than that, they are wider, so their length is more than their width. Most of the bones of the upper and lower limbs are long bones, and the short bones, they are about as broad as they are long. The short bones, they are having same length and width, and they are nearly cube shaped or round and are present in wrist and ankle like the carpals and tarsals. The carpals of the wrist and tarsals of the ankle. And then there are flat bones. Flat bones are the bones that have a relatively thin flattened shape and are usually curved. These bones are like skull bones, the ribs, and the breastbone, that is sternum, and the scapula, that is the shoulder bone. And then we have irregular bones. Irregular bones are the bones that don't have any irregular shape. They are bones of the face and the vertebrae, so they are not uh, having a proper shape. Long bone has three distinct structures, that is a diaphysis or a shaft of the bone, and then the epiphysis, and then there is the epiphyseal growth plate. This epiphyseal growth plate, in case of mature bone, is converted into the epiphyseal line. So the diaphysis or shaft of long bone is composed primarily of compact bone, which is mostly bone matrix with a few small spaces. So here in the shaft, the spaces are very less between the collagen fiber, and the uh, spaces are filled with calcium and phosphate. And then the epiphyses are the ends of bones. They are made up of spongy bone or cancellous bone. Cancellous or spongy bone is the bone that has the spaces between its fiber or the bone matrix, extracellular matrix. So the here we can see it is the end of the bone or epiphysis, so it is spongy, while diaphysis is compact bone. It is not having any spaces. So uh, the the epiphysis at the near joints, it is in connection with articular cartilage or haline cartilage. So the epiphyseal growth plate, here it is the epiphyseal line. So this epiphyseal line, in case of the growing bone, it is epiphyseal growth plate. So this epiphyseal growth plate is haline cartilage located between the epiphysis and the shaft of the bone or diaphysis. So growth in bone length occur at the epiphyseal growth plate, but when bone stops growing in length, the epiphyseal plate becomes ossified, and then it is called as epiphyseal line. So here haline cartilage was present when this bone was growing. So this haline cartilage was dividing. It is the haline cartilage cells, chondroblast, they were then dividing and making more bone toward epiphyseal side and toward diaphysis. So these cells later on were converted into osteoblast and osteocytes. So later on, when this person got fully mature or adult, at that time this bone stops growing, and this epiphyseal, the cells of the cartilage here, they also converted into osteocytes, and they are now trapped into in the calcium and phosphate. So these cells, they are not growing now, so that is called as epiphyseal line. So the length of the bone is also not growing. So the here spaces are present in the spongy bone or the epiphyseal bone, but on the crown side or the cortex side, there in diaphysis there are no spaces between fibers. It is compact bone, but there is a large cavity in diaphysis. This cavity is called as medullary cavity or medulla. Medulla means central cavity. So this cavity of bone, they are filled with bone marrow. That is the spaces here in spongy bone, they are filled with bone marrow, and then this medullary cavity, it is also filled with bone marrow. So these bone marrow could be red bone marrow or they could be yellow bone marrow. Red marrow is for the formation of blood cells, and yellow marrow is actually adipose tissues. In children, these spaces are filled mostly with the red marrow, and they are producing a lot of blood cells. But when children get mature, yellow marrow comes in place of the red marrow, and the red marrow is only located at the near joints. So it is only present in the uh, bones of skull and limbs, and uh, these proximal areas, they have the areas that are near to joints, they have red bone marrow, but rest of the long bones, they are filled with fats or yellow marrow. Like cartilage, the bones, they are also covered by a membrane of connective tissue that is called as periosteum. Periosteum mean peri mean along and osteum mean bones. So this membrane of connective tissue is uh, making outer surface of the bone. The outer fibrous layer is dense, irregular collagenous connective tissue that contain blood vessel and nerves. So the blood vessel and nerves, they are running in periosteum. So periosteum is like a double layer. The outer fibrous layer is dense. It is having more fibers that are compactly bound, irregular collagenous connective tissue that contain blood vessel and nerve. The inner layer is single layer of bone cell which include osteoblast, osteoclast and osteochondral progenitor cells. Osteoblasts are to produce the bone, that is extracellular material, and osteoclasts are to remodel or chew the bone, and osteochondral progenitor cells are the cells that are responsible for the formation of these osteoblasts. They are actually the stem cells, and the here where tendons and ligaments, they get attached to bones, they become fused with the periosteum, become part of the periosteum. So they strengthen the attachment of the bones with tendons or ligaments. The endoosteum is the membrane connective tissue that is lining the medullary cavity from inside, and these endosteum, they are single layer of cell which include osteoblast, osteoclast and osteochondral progenitor cell. Here we have to keep it remember that the chondroblasts, they are present only in the epiphyseal uh, growth plate where the bones grow in length, and then the osteoblast, osteoprogenitor, control progenitor cells, osteoclast, they are only present in the inner layer of the periosteum, that is the membrane that is covering bones from outside, and then they are also present in the endosteum that is covering the medullary cavity or the bones from inside. So the bone grow in diameter by the help of these cells in endosteum, and the periosteum, and bone grows in length only at the epiphyseal growth plate by the help of the cartilage here.
By doing histology, we can identify two types of bones on the basis of their structure, that is woven bone and lamellar bone. Woven bones are weak, they are immature bones, and their matrix is interwoven collagenous fibers, and these the osteoclasts, they are randomly present in the matrix, and the woven bone is replaced by lamellar bones at 2 to 3 years of age, and these fibers, they are not in the form of sheets in the woven bone, so it is giving them less strength because fibers are arranged haphazardly, so they are not giving strength in one direction. Lamellar bone, they are strong bone, they are mature bone, they are strong because their fibers or the matrix is in the form of sheets or layers. So these layers, they give strength in one direction. So these layers, they are called as lamina. So most of the mature bones, they are in the form of lamellar bones or strong bones. In generally, the collagen fiber of one lamina are parallel to another, but at an angle to the collagenous fiber in the adjacent lamina. So they give strength in the strength to the bone. And the osteocytes, they are present within their vacuna or arranged in layers that are sandwiched between lamina. Bones, either they are woven or lamellar, they can be classified according to the amount of bone matrix relative to the amount of space present within the bone. Means how much bone matrix is present, how much spaces are over there. So cancellous bones, they have less bone matrix and more spaces than compact bone, which has more bone matrix and less spaces in the compact bone. Cancellous bones are made up of interconnecting rods or plates of bones, and these rods or plates, they are called as trabeculi. Between the trabeculi are spaces that are filled with bone marrow and blood vessels. Here we can see. So cancellous bone is sometimes called as spongy bone because they are porous. They have pores. They are the trabeculi. They are thin, 50 to 400 micrometer, and they are consist of several lamella of the that are packed with osteocytes, and each osteocyte is associated with other osteocyte through canaliculi. We'll see them later on. Canaliculi are actually gap junctions that are for the exchange of material between cells, because we know the in lamellar bone or mature bones there is extracellular matrix in which the cells are trapped. So in order to exchange material, they need some special type of connection. These connections, they are called as canaliculum. The blood vessels they don't penetrate in the trabecular, so osteocytes they need canaliculi for exchange of material. The surfaces of trabeculi they are covered with a single layer of cells consisting mostly of osteoblasts with a few osteoclasts. So the trabeculi in spongy bones they are arranged in the line of stress, and if the stress angle is changed a little bit, then they uh, trabeculi they start to grow toward that new line of stress. The compact bones they are denser because they have more matrix, so they have less spaces than cancellous bones. Blood vessels enter the substance of the bone itself, and the lamella of compact bone are primarily oriented around those blood vessels. The lamella they make a certain type of circular structure around blood vessels. Vessels that run parallel to the long axis of the bone are contained within the central or Haversian canals. I will see them later. So the central canals they are lined with endosteum and contain blood vessels, nerves, and loose connective tissue. So the compact bones are more strong as compared to these spongy bones. We see the structure of these bones in the next slide. As we know, the spongy bones they are deep bones and they have more spaces between their structure and matrix, and the lamella of these bones they are not arranged in the form of osteon. There are no central canals, no blood vessels in central canal, and the nutrients they diffuse from one layer to another layer through canaliculi. Here we can see structure of the spongy bone. It is in the form of trabeculi or the pillars. So these pillars they are arranged in different directions as we described them previously. So these pillars they are made up of lamella of the bones. These lamella they are making a circular structure, but they don't have a central canal and blood vessels. So the cells in these lamella they are, here we can see an osteocyte. It has many extensions that are arranged with the aligned with the other cells. They are in contact with the other cells through gap junction, and these osteocytes they are present in the lacuna or depressions, and then these osteocytes they get nourishment through these canaliculi. So these bones they cannot bear a lot of weight, so they are only present where the muscles they get attached to the bone. So they are present on the ends of the bone or at the faces of the bones, and they are mostly found in flat bone, joints, and skull. The trabeculi or the pillars they may support or protect the bone marrow. The methods of the spongy and the compact bone are same, but there is difference in the arrangement of the canaliculi, the fibers, and the blood vessels. So the compact bones they are denser and they have few spaces than cancellous bone, as we know already. So blood vessels they enter into the compact bone. So the matrix of the bone in the form of lamella or the layer is arranged along the blood vessel that enter into the bone. So the blood vessel they run through a central canal. So this central canal or Haversian canal, uh, that is called as Haversian canal. So the blood vessels they are running in that and nerves and new connective tissue. They are also present here. And the concentric lamella. Concentric means they are sharing same centric center. So these are the lamella, these are the layers. So these layers they are making a structure that is this whole structure is called as an osteon. Osteon is made up of blood vessels, nerve, the endosteum that are present in the central canal are lined by the matrix of the bone in the form of concentric lamella. This whole structure is called as an osteon. Osteon is the basic functional unit of the compact bone. So osteocytes they are located in lacuna between the lamellar rings. So the lacuna are the depressions where the matrix is not present and cells are present. And the canaliculi are radiating between lacuna across the lamella, producing the appearance of minute cracks across the ring of the target. So these are the canaliculi that are here. You can see these radiating structure here. Here you see them. So they are passing from one lamella to another lamella. So canaliculi of one lamella, they have connection with the canaliculi of another uh, lamella. So these canaliculi are actually extension of the osteocyte. So osteocytes in one lamella has connection with the osteocyte in another lamella through these canaliculi of the branches. So there are gap junction between these. And we know gap junctions are the bridges for exchange of material between cells. So the inner cells that are near to the blood vessel they get food and nutrients and other gases, oxygen. So these cells they share these nutrients and gases with the cell in the other layers of the lamella that are away from the blood vessels. And similarly, the waste material they are passed from the outer layers to the inner layer cells or osteocytes, and then from the inner layer to the internal layer that is within contact with the blood vessels to pass the waste material back to the veins. So the outer surface of compact bones are formed by circumferential lamella. Here you can see the internal structure is made up of concentric lamella. Concentric means they are sharing same center. They are in circular fashion. But outer lamella, they are not in the form of circle. They are linear. They are flat. So the circumferential that are on the circumference of the bone, they are flat plate. And some bones like the bones of the face that are the flat bones, they even don't have any osteon in them or concentric lamella. They are entirely made up of circumferential lamella. So here you can see in between the concentric lamella or circular lamella, there are uh, interstitial lamella. These interstitial lamella, they are actually the remnant of concentric or circumferential lamella that were being modeled during bone development. So these are the remains of previous circular or the circumferential lamella. These are called as interstitial lamella. Osteocytes in the bone, as we said previously, they receive nutrients and eliminate waste products through the canal system within compact bone. So this is the central canal system through which they receive nutrients. As we know, there are blood vessels, so they receive nutrients from here and dump their waste back into these blood vessels. So blood vessels how they enter into the bone. They are coming from the periosteum. Here we can see the fibrous layer of periosteum, and then is the cellular layer of the periosteum or inner layer of the periosteum. We can see blood vessels that are running outside of the outer layer or fibrous layer of periosteum. From here, the blood vessel enter into the the blood vessel. They also can come from medullary cavity or the central canal. Here you can see the medullary cavity or the central canal of the bone. So the blood vessel they also enter from here. So how they enter into the osteons or central canal is through perforating or Walkman's canal. So these are the Walkman or the perforating canal. Here we can see from periosteum and from endosteum from medullary cavity. They enter through the canals that are at right angle to the length of the bone. Bone length is here and they are at right angle to or perpendicular to the bone length. So these perforating canals they allow the blood vessel to enter into the circular canal. So perforating canals they are not surrounded by concentric lamella, but they are crossing the concentric lamella. So they are at right angle to the concentric lamella and they are entering into the central canal. And in central canal, they receive blood vessel and from perforating canal, and then, as we said previously, nutrients in the blood vessel enter into the central canal, and from central canal, it is passed to the canaliculi of the osteocytes that are near to the blood vessel into the innermost lamella. And from the canaliculi of these osteocytes, the nutrients and food enter oxygen they enter into the osteocyte in the next lamella and so on till to the osteocyte into the outer layer of the bone matrix. And similarly, the waste material they go from the osteocyte of the outer layer to canaliculi into the osteocyte of the inner layer and so on until it reaches to the innermost layer of the lamella, and they are their osteocyte they have direct connection with the blood vessel to release it back into the blood vessel. The things that we have to keep remember that an osteon is basic functional unit of compact bone, and the bones they are very strong from top to bottom because the osteon they are in parallel fashion, so they give strength in one direction. These bones they are weak from side to side. If pressure is applied perpendicular to the length of the bone, then these bones they cannot withstand that pressure and they could be broken. So they are only strong from top to bottom. So what is the osteon? It is the Haversian system in the compact bone. Is the osteocyte in concentric layer around the central canal are present. So concentric layers are lamella and central canal is Haversian canal. So this whole structure is called as Haversian system. In compact bone they make the diaphysis of the long bone where there is the shaft of the bone and they have a central canal or medullary cavity. The ossification or osteogenesis is the formation of bone. So these in fetus during development, bone formation occurs in two patterns called intramembranous or membrane ossification and endochondral ossification. The terms describe the tissue in which bone formation takes place. Intramembranous ossification, the connective tissue membrane, and endochondral ossification in cartilage. Chondral means cartilage. So both method of ossification initially produce a woven bone and then it is remodeled into the lamellar bone or the mature bone. Electrical connective tissue is in the form of endocrine and the mucous connective tissue is only present in the lysocord and the mesenchymal connective tissue gave rise to all kinds of connective tissue membranes presently. So in case of intramembranous ossification, after membrane ossification, these mesenchymal cells they start to collapse on the vacuum base in form of membrane. So this mesenchymal membrane it contains mesenchymal cells that uh, start to be converted into osteochondral coordinator cells. We know osteochondral coordinator cells are the structure of both the cartilage and the bone. So these osteochondral coordinator cells here in the membrane ossification, they start to have the osteoblasts, and these osteoblasts they uh, they, they start to produce the matrix of the bone. So these osteoblasts they get trapped in the matrix and they are now called as osteocytes. So these osteocytes they give rise to tiny radiculi of human bone. And then these tiny radiculi they are strengthened and they are strengthened by more osteoblasts rather than more matrix of the bone so that the radiculi will be able to become larger and larger. So these radiculi they join together and here you can see one radicular ventriculus. So many radiculi they join together to make the spongy bone. So here there are many radiculi. So these are making essentially a spongy bone. A spongy bone has many radiculi that are separated by spaces and these cells within these spaces they start to make red bone marrow. So the cells along the bone they start to get decay in the form of periosteum. They become specialized and they make periosteum. So the osteoblasts in the periosteum they start to make a layer of complex bone that is present on the outer surface of the spongy bone. So the end product of this intramembranous ossification are the bones that are compact at the outer surfaces and they are only cancellous in the inner side. Later on, these radiculi they are because radiculi are arranged heptagonally, so they are weak bones, they are immature bones. So later on, they are these radiculi they are replaced by the parallel layers of the lamella and they make the lamellar bone. The membrane ossification occurs in the bones of skull, the lower jaw, and the diaphysis are the central shaft of the collarbone or clavicle. So these bones they have center of ossification. In mesenchymal membrane there is formation of a center of ossification. So bone that is formed within the center of ossification is the older bone and bone that is around the outer surface of the neural bone. So the neural bone, they are then they are not that occupied. So in case of the larger bones of the skull bone, they are their outer surface or the outer layers, they are not occupied at first. So later on they get occupied and they get their formation of integrity. Endochondral ossification is also called as replacement bone. In endochondral ossification, first the cartilage is formed and then this cartilage is converted into bone. For the formation of cartilage, the mesenchymal cells of the future connective tissue, uh, they aggregate and they have the point where the future bone has been formed. Mesenchymal cells, they are converted into chondroblasts, and these chondroblasts, they give rise to the cartilage, and they release the cartilage matter, that is the collagen fiber, and they get trapped into that cartilage to become the chondrocytes. The cartilage is surrounded by perichondrium. So the cartilages, they are surrounded by a constitution that is called as perichondrium. So perichondrium is not only present where the joints have to be formed by bone, and the blood vessels, when they invade into the perichondrium, uh, the osteochondral progenitor cells within the perichondrium, they become osteoblasts. Previously they became chondroblasts. Now when blood vessels enter, they become the osteochondral progenitor cells. They become, because of the perichondrium is a membrane that contains osteochondral progenitor cells. So osteochondral progenitor cells, they have potential of formation of both osteoblasts and chondroblasts. Previously, when blood vessels were there, they became chondroblasts. Now, when blood vessels enter, they have become osteoblasts. So these osteoblasts, they produce compact bone on the surface of the uh, cartilage, and that form of bone collar. So this formation of bone collar in the form of compact bone. At the same time, the model, that is the baseline of the cartilage, it grows in size, and the cells in the center of the cartilage, they increase in size, and the matrix, it becomes mineralized between the calcium and phosphate. They get deposited into uh, the calcium in the form of calcium carbonate that is put into the matrix. So that cartilage is called as calcified cartilage. So the chondroblast, they die here. So the chondrocytes, they die, and there are var lacuna in the bone. So the blood vessel, they grow in the leukemia, and connective tissue surrounding these blood vessels, they build more osteoblasts, and osteoblasts from the periosteum. Osteoblasts, they produce bone on the surface of the calcified cartilage, forming bone pediculi, which changes the calcified cartilage of the diaphragm into tensionless bone. This area of bone formation is called the primary ossification center. So primary ossification center in long bone is in diaphragm. So In long bones, the scanty ossification center is also found in epiphysis, and here the cartilage it starts to produce the osteoblast, and these osteoblasts they lay down in the tissue in the form of bone. In fact, all the bone is then replaced with all the cartilage is replaced with bone, except that the epithelial plate, where there is cartilage present and it helps to grow the long bone until the adulthood. In adults, this epithelial plate it precisely it becomes mature and it has no more the chondroblast or osteoblast, so it becomes epithelial line. So bone in adults cannot increase in length because the epithelial plate has been converted into the epithelial line. Endochondral ossification is also known as the replacement bone, and it occurs in all of the bones except the skull and the diaphysis of the clavicles and mandible. So all the other bones they originate through endochondral ossification. The bones grow in both directions, that is lengthwise and widthwise. At lengthwise it grows at the epithelial plate. So the epithelial plate is organized into four zones. The zone of resting cartilage, that is at the end of the bone or to the outer side of the epithelial cycle. And this zone of resting cartilage, it is contains randomly arranged chondrocytes that do not divide rapidly. Then there is a zone that is called a zone of proliferation or proliferating cartilage. These are the cells that are producing new cartilage in interstitial cartilage layers. The chondrocytes are divided and form a column with the main stacks of layers of from in zone of hypertrophic cartilage or zone of hypertrophy. The chondrocytes produce in the zone of proliferation that mature and enlarge. So these zones of hypertrophy they have enlarged size of cartilage that have been chondrocytes. Then zone of calcification. Zone of calcification is a thin zone between the diaphysis and the epiphysis, and this zone is made up of cartilage matrix that is dendrite with calcium cartilage. So the hypertrophy cartilage chondrocytes they die and blood vessels from diaphysis grow into the area. So the calcification starts here. That is the laydown of calcium cartilage. The bones grow lengthwise and widthwise, that is increasing length and diameter occur. The lengthwise, the long bone they grow at epithelial plates only, and the epithelial plate are actually made up of cartilage. So that cartilage it grows in the form of a positional growth and interstitial growth, both growths. Like in cartilage grows by these both growths. A positional growth is the growth in which new tissues are laid down on the previous tissue. So the thickness of the tissue increases. And interstitial growth is by dividing cells that are present within the tissue already. So these cells they divide and the bone grow in size or cartilage grow in size and later on ossification occurs. So the width wise because it is the mature bone So the bones they can grow only by oppositional growth. It means new layers of matrix is laid down on the previous matrix. So the cells within the bone they are already mature, so they cannot divide and make new cells of the matrix. These osteocytes they can only maintain them. So oppositional growth and interstitial growth we have to make the concept clear that oppositional growth is formation of new tissue on the previous tissue, and interstitial growth is formation of new cells by cell division within the already present tissue. They occur within the tissue. So in epiphyseal plate, because there is cartilage, so these cells they have potential to grow by oppositional strategy as well as interstitial. But the in case of weight wise only, osteocytes and osteoclasts they are present, so they can osteoclast they can lay down new bone on the previous bone. That is called as oppositional plate. As you know, the compact bone they are made up of osteon. So if the diameter of the bone increases, it is by increasing number of osteon. So how the osteon are made? We know the bones they are lined by periosteum, and the periosteum is a double membrane, that is the fibrous outer layer and then the inner cellular layer. And we know the blood vessels they are running on the outer fibrous layer, and then we also know that these in center of the compact bone there are osteum, and on the periphery there are the circumferential lamella that are flat. So in these flat lamella, the blood vessels of the periosteum they start the periosteum it start to dip down inside the toward the center of the bone, and the ridges are formed, and then these ridges of the periosteum they get fused with each other, and so the blood vessels they get trapped between that was previously present on the surface of periosteum. Now they have become part of the inside of the this whole structure, that is osteon-like structure. So now the periosteum that is enclosed by this cavity is called as the endosteum, and the blood vessel they are running through a canal, that canal is called as the uh, central canal or Salvation canal, and now a new osteon is formed. So by the formation of new osteon, the diameter of the bones are being increased.
Like other living tissues, if bone get damaged, it undergoes repair, and the repair process is composed of four uh, processes. Number one is formation of fracture hematoma. We know the blood vessels they are going into osteon, into the central canal and the perforated canals, and if the bone get damaged, these blood vessels are broken, and the blood comes into the tissue, and where it get clot, and this clot is called as hematoma. So this hematoma hinders the supply of nutrients to the osteocyte on vicinity. So these osteocytes they die, and there occurs of an inflammation, and there is hindrance to the function of this bone. Or swelling also occur at the uh, spot of the fracture or the breakage. After formation of hematoma we said is a clot. After formation of this clot, there is formation of a callus. So there is formation of two types of callus, an internal callus and external callus. So these, what is a callus? Callus is a tissue that forms at fracture site, and it connects the broken edges of the bone. So it occurs from inside and from outside. And when this clot was formed, after a few days of clot formation, the blood vessels they again grow into this clot. The blood vessels growth is called as angiogenesis. So these blood vessels through a process of angiogenesis comes into the clot, and this clot is removed by the activity of phagocytic cells. The osteoclasts they break down the damaged edges of the bone, that are the dead bone uh, particles. They are chewed by osteoclasts here. The fibroblasts here they produce the fibers, collagen fiber and other extracellular material, and that tissue is called as called as granulation tissue. The fibroblasts they keep on laying the fibrous tissue. So this fibrous tissue is made up of collagenous fiber, and it can hold the broken edges of the bone. So this fibrous tissue is called as a callus. Chondroblast in the perichondrium and the periosteum and the endosteum. These chondroblasts they uh, they start to lay down cartilage in the uh, broken bone area. Then the osteochondral progenitor cells in the endosteum and the periosteum they become osteoblasts, and they produce new bone, and that contributes to the callus. So these osteoblasts they lay down the extracellular material, that is of calcium and phosphate ions and collagen fibers. So the ossification of the cartilage takes place like in the development of the bones that we saw in previous slides, that the cartilage are converted into bones. So same process, the osteoblast they convert this cartilage into the bone by the process of ossification. So a cancellous bone is formed at the fracture first. Then this woven bone is converted into the lamellar bone, because the lamellar bone is the strongest bone. So repair is not complete until the woven bone is converted into lamellar bone. This conversion of the woven bone into lamellar bone is called as bone remodeling. We saw formation of new osteon in previous slide. So with the same mechanism, remodeling of the woven bone takes place. Bones are part of skeletal system, as well as they take part in regulation of calcium levels in the blood, and they act as storage of calcium and phosphate. The bones, when there are low calcium levels in blood, the calcium comes from bone and regulate the or maintain the levels of calcium in blood. And if calcium levels become high in blood, then these calcium they are stored or deposited into the bones. So when this whole mechanism is regulated by two hormones. A hormone that is the parathyroid hormone comes from parathyroid gland that is present on the rear lobes of the thyroid gland, and the other hormone that is calcitonin, it is released from the thyroid gland itself. And we know there are follicular cell in thyroid gland that release T3 T4, and there are parafollicular cells near to follicles or outside of follicles. These cells, they release calcitonin. So when there are low calcium level in blood, parathyroid hormone is released. This parathyroid hormone increases the number of uh, osteoclasts. So we know the osteoclasts, they chew the bone. So osteoclasts, they start to chew bone, and they chew the um, calcified structure of the bone. So calcium is released into the blood, so calcium level is increased. Parathyroid hormone also increases calcium level of the blood by reuptake of calcium from nephrons and uptake of calcium from the uh, intestine, and it also helped to increase the levels of vitamin D. So vitamin D helps to increase the uptake of calcium from intestine. So the parathyroid hormone is actually, in case of bones, it is increasing activity of osteoclast, and it is increasing the formation of osteoclast. And if calcium levels are increased in blood, then parathyroid hormone is not released or its levels go down. So the osteoclast activity is decreased. So there is no more damages to bone and no more calcium is being released into the blood. So calcitonin that is released from thyroid gland decreases the osteoclast activity, and it an increase in blood calcium level increases the uh, calcitonin that inhibit the osteoclast activity. So the parathyroid hormone and calcitonin, they may act in an antagonistic action. So here in this diagram we can see that if blood calcium levels are low, then the parathyroid gland, it has a receptor, it detects the calcium levels, so it releases parathyroid hormone. So these parathyroid hormone increases the osteoclast activity for bone resorption or bone damaging, and they also increase kidney reabsorption of calcium and from intestine, so formation of vitamin D. So that increases, all these activities increases the blood calcium level. So this give feedback to the uh, parathyroid hormone gland to not release more uh, parathyroid hormone.